objectives are: number one is the phospholipid. First, we will discuss about the classification of phospholipid. There are major two classification: glycerophospholipid and sphingophospholipid, and their synthesis of glycerophospholipid and degradation of glycerophospholipid. Similarly, we will discuss about the synthesis and degradation of sphingophospholipid. After that, the second main objective of today's lecture is what is surfactant, what is their role, how is synthesized, what are their major functions of surfactant, and the law of Laplace, which is actually apply on the pulmonary alveoli. The third things which we will discuss about the respiratory distress syndrome. So there are two types of major. Today we will discuss mainly focus on neonatal respiratory distress syndrome. So we will start with this case of neonatal respiratory distress syndrome. A 30-year-old pregnant mother, G3P2, is in her 30 weeks gestational, delivered 1.5 kg baby girl, cesarean section done. After some complication in pregnancy, the mother received one dose of beta-methazone. This is one of the glucocorticoids 10 hours before delivery. The baby was resuscitated, intubated, and her surfactant administered in the delivery room within 15 minutes of delivery. uh, Baby Apgar score were 2, 5, and 6 at 1, 5, and 10 minutes. The baby was mechanically ventilated for severe respiratory distress syndrome. So there are some of the point which we have to be remember. This baby was delivered at 30 weeks, normally more than 38 to 40 weeks, between 38 to 40 weeks. So it's a premature baby, and they have received, this mother received the beta-methazone is glucocorticoids. What is the role of glucocorticoids in this case? And we have also discussed why this surfactant is given in this case, and why this baby goes into the mechanical ventilation. This we will discuss at the end. First we will discuss about phospholipid and surfactant. So phospholipids are the compound lipids. They are called compound lipids because they contain one of the special group, the phosphoric acid. That's why they are called the phospholipids. They are the lipids. They are the compound lipids. They containing the phosphoric acid in their structure. The any lipids have fatty acid as well as fatty alcohol, but when they are called compound lipid, when you call the compound lipid, the fatty acids and fatty alcohol combine with phosphoric acid. It's called some of the compound is attached as a phosphoric acid and one special nitrogenous compound. This nitrogenous compound actually further classify these phospholipids. The nitrogenous compound may be the choline, serine, ethanol, amine, and the fatty acid is always there in this phospholipids, and fatty alcohol is there. If the glycerol is attached, it is called glycerophospholipid. If sphingosine is attached alcohol, then it's called as the sphingophospholipid. So as I told you, they are further classify glycerophospholipid and sphingophospholipid. If glycerol is attached, it is called the glycerophospholipid. If sphingosine is attached, it's called the sphingospholipids. So they have the glycerol, but they have sphingosine in sphingophospholipid. They have two fatty acids. Similarly, they have one fatty acid, sphingolipids, phospholipids, and they have phosphoric group. They also have a phosphoric group. They have some alcohol amine like choline, serine, ethanol amine. Similarly, they have sphingophospholipid always contain the choline. So this is the further classification of phospholipid. As I told you, the phospholipid can be classified the glycerophospholipid and sphingophospholipid. On the basis of glycerol and sphingosine, they are classified the glycerophospholipid and sphingophospholipid. But glycerophospholipid are further classified on the phosphoric acid, lecithin, cephalin, phosphatidylserine, and phosphatidylsphingosine inositol. This is actually classified on the basis of nitrogenous basis. If choline is attached, it is called lecithin. If ethanol amine is attached, it is called cephalin. If serine is attached, it is called the phosphatidylserine. And the sphingophospholipid, as I told you, they contain the sphingosine as an alcohol, and that's why they are called as sphingomyelin as well. They are found in all cells, milk, egg yolk in the form of lecithin. So this is what I've told you. This always the phospholipid contain one glycerol. If they contain glycerol, they are called glycerophospholipid. They are the two fatty acids, the acyl group, acyl residue. They have phosphate in it, and they have some one of the nitrogenous amine. This, if the choline is attached here, it is called as the uh, phosphatidylcholine or lecithin. If ethanol amine is attached instead this nitrogenous base, then it is called as the this phosphatide is called as the lecithin, or phosphatyl ethanol amine is also called. If the serine is attached, it is called phosphatyl serine, and if its anisotol is attached, it is called as phosphatyl anisotol. So this is the glycerophospholipid because they have a glycerol in it, and phosphate group, that's why they are called phospholipid. And this compound is also have a phosphate group. They also have a one acyl group or the fatty acid, and but they contain alcohol, not the glycerol, but they contain the sphingosine. So that's why they are called the sphingolipid. And they have amine sugar, the choline. Always choline is there in sphingophospholipid. So there are two classes: the glycerophospholipid, the sphingophospholipid. And glycerophospholipid can further classify on the basis of this amine alcohol sugar. It can be the choline, that's called lecithin or phosphatidylcholine. If ethanol amine is attached here, then it's called the phosphatyl ethanol amine, or it's also called as a cephalin. If serine is attached, it is called a phosphatyl serine. If anisotol is attached, it's called a phosphatyl anisotol. This is the classification which I told you. This is the fatty acid, two fatty acid, one glycerol, one phosphate, and there one compound. Choline is attached, it's called the lecithin. If ethanolamine is attached here, then we can call it as a cephalin. Can be attached. Serine is attached, we call phosphatyl serine. So the most common glycerophospholipids we will discuss today, the five major one, and how this glycerophospholipid synthesized. Then we will discuss about the sphingophospholipid and its synthesis and degradation. First, we will discuss about the simplest one, phosphatic acids. It does not have a specific function. The second one is the lecithin. It's formed the glycerol, two fatty acids, phosphates, and choline, which is the nitrogenous base. It is a phosphatic acid, choline, and it's called the phosphatyl choline. So the nitrogenous base only when changed with glycerol, two fatty acid, and phosphate. The nitrogenous group is choline, is called lecithin. If this instead of choline, this ethanolamine is attached, it's called as a cephalin. I will show you. So, the lecithin is the second one, which is also called phosphatidylcholine. It's the most abundant group of phospholipid in cell membrane, and they are storage coming of this choline coming from the donor or methyl group in methylation reaction of acetylcholine in nerve transmissions, and the dipalmitylcholine or dipalmityl lecithin, which is mainly maintain the surface tension, and which is the major topic of today lecture, where we discuss about this dipalmitylcholine, which maintain the surface tension and produce the surfactant. And this surfactant actually try to prevent the collapse the alveoli. So dipalmitylcholine is the major component which form the surfactant. Of the deficiency of surfactant, then the respiratory distress syndrome in infant can occur. So now come back to the classification. The phospholipid in cephalin is a chemical structure. As I told you, instead of choline, there's replaced by ethanolamine, and this cephalin is mainly used in clotting factors, which produce the thromboplastin, and the major function in clotting of blood as well. Whereas the fourth classification of glycerophospholipid is phosphatidylserine, and phosphatidylserine is an important phospholipid which supports cell integrity, found in high concentration in brain. And it is essential for normal neuron structure and function, and may play a critical role in maintaining concentration and memory. The last one is the phosphatidylinositol. It's a major function for the second messenger systems. It is one of the cell membrane lipids and it's play a role in cell signaling, mainly this phosphatidylinositol. So look at this, and this is the glycerol part, the two fatty acid, the phosphate, and the compound, nitrogenous compound is the inositol. No need to remember the structure, but the function you have to be remember. How this glycerophospholipid is synthesized? So they were mainly synthesized in the liver. Actually, there are major three steps of synthesis of glycerophospholipid. Number one, the glycerol is activated by ATPs and they given their glycerol 3 phosphate. The ATP given their 3 phosphate and it's changed. This glycerol is converted into glycerol 3 phosphate. And now the two fatty acid come, the fatty acyl group come and give this fatty acyl group to this glycerol 3 phosphate. When they give their glycerol 3 phosphate and they convert into the phosphatic acids actually. Okay, so after that the choline and ethanol amine, it depend upon the lecithinous formation occur or the cephalin formation occur. If lecithin occur, the choline will occur. Choline will come to combine this phosphatic acid, or otherwise the ethanol amine will come and combine with this phosphatic acid and form the lecithin or cephalin. I will show in diagram as well. And this process is mainly activated by cytosine triphosphate and which convert into the CDP choline and CDP ethanol amine. So this is the process how the phosphatic acid synthesized, the lecithin and the cephalin is synthesized. So mainly, as I told you, the glycerol convert into glycerol 3 phosphate with the help of glycerol kinase and ATP. ATP give their phosphate and form into the glycerol 3 phosphate, the third part where the phosphate is attached. So this glycerol 3 phosphate then combine with two fatty acyl groups, and this two fatty acid when combined with this 3 glycerol 3 phosphate, they form the phosphatic acid, and this is the simplest form of glycerophospholipid. And this phosphatic acid and further convert into the diacyl glycerol and remove their one phosphate and form the diacyl glycerol. And this diacyl glycerol can combine with ethanolamine to form phosphatyl ethanol or cephalin, or they combine with choline and they form phosphatyl choline, which is also called the lecithin. So I'm repeating this one. This glycerol come to convert in glycerol 3 phosphate. Remember this dihydroxyacetone, which is the one of the intermediate of glycolysis, which can also come to form the glycerol 3 phosphate. And this glycerol 3 phosphate when combined the two fatty acyl group or two fatty acids, and they form the phosphatic acid, and phosphatic acid then further convert into diacyl glycerol, and this diacyl glycerol can combine with choline as well, can combine ethanol amine, and this choline or ethanol amine, if they combine with this diacyl glycerol combine with choline, they form the phosphatyl choline. If they combine with ethanol amine, they form the phosphatyl ethanol amine. The cephalin is this lecithin is formed here, and cephalin is synthesized here. So the how this phosphatidylserine is formed? The phosphatidylserine is formed when phosphatyl ethanol amine. This is this one. This phosphatyl ethanol amine is react with serine. So look at this. When phosphatyl ethanol amine is react with serine, which replace the ethanol amine moiety, which replace the ethanol amine moiety and form the phosphatidylserine. So this is the how this phosphatidylserine is formed. How this glycerophospholipid are degraded? There are four major types of phospholipases enzymes which help in degradation of glycerophospholipid. Number one is phospholipase A1, phospholipase A2, phospholipase C, phospholipase D. Phospholipase A1 is actually break down the first fatty acids and convert this glycerophospholipid into lysolecithin or lysocephalin. It depend upon this nitrogenous base. So the first phospholipase A1 when break down the first fatty acid, the second phospholipase A2 break down the second fatty acid. How this degradation occur? I hope you are understanding. The first is the phospholipase 1, which break down the fatty acid 1, and the second phospholipase 2 break down the fatty acid 2, and the phospholipase C is mainly broke down this the glycerol and phosphate binding, and this break down this glycerol and this phosphate and nitrogenous base in one place, and then this last the phospholipase D actually mainly break down this bonding for phosphodiesterase bond between this phosphate and the nitrogenous base. So these are the way how this phospholipases break down this glycerophospholipid. And there is a very important, this phospholipase 2 have some other important function which we will also discuss here, and the phospholipase C. So as I told you, the phospholipase 1, which act on the ester bond in position 1 and remove the first fatty acids and form from the lecithin or cephalin and produce lysolecithin or lysocephalin. Then the second phospholipase A2, which act on the second position and remove the second fatty acids. And phospholipase C, which is phosphodiesterase, act on the link between the glycerol and phosphate and giving the free glycerol and phosphate plus bases as well. And finally the phospholipase D, which is phosphatase, also called phosphatase, which actually link between the phosphate and base and giving. So this is how there's a phospholipase 1 breakdown first, 2 2, and third one is phospholipase 3 breakdown here, and phospholipase D. So phospholipase A2 has very important role, so we have to discuss here. So act on the membrane phospholipid. It actually, when the phospholipase A2 break down, it release arachidonic acids, and this arachidonic acids is very important for synthesis of some of the prostaglandin and leukotrienes, which causes the inflammations. So this pathway is catalyzed by cyclooxygenase, and this enzyme, which is mainly inhibited uh, by aspirin, and corticosteroids can inhibit the phospholipase A2. So cyclooxygenase pathway or lipoxygenase pathway, there are two pathways, and this is mainly produced after the breakdown of arachidonic acids. So arachidonic acids entered into the synthesis of different prostaglandin and leukotrienes, which causes inflammation, and this pathway is catalyzed by cyclooxygenase enzymes, and this enzyme is inhibited by aspirin. Okay, and corticosteroids is mainly directly inhibit the phospholipase A2 as well. So phospholipase A2s are commonly found in mammalian tissues as well as some insects and snakes as well, and increase the presence and activity of phospholipase 2 resulting from snake or insect bite. Arachidonic acid is released from the phospholipid membrane as a result of inflammation and pain at the site of insect bite. So look at this cell membrane phospholipid. Phospholipase A2 break down this arachidonic acid into two pathways, the cyclooxygenase pathway, the lipoxygenase pathway. And as I told you, this glucocorticoids directly inhibit the phospholipase A2, whereas cyclooxygenase is mainly inhibited by NSAID or aspirins as well. So now the second is phospholipase C. After phospholipase A2, now what is phospholipase C? When phospholipase C is, it's release the inositol triphosphate and diacylglycerol, which is very important in the degradations in the second messenger system. So it's important is very whenever the phospholipase C act on the phosphodiol inositol biphosphate, it release inositol triphosphate and diacylglycerol, which are further importance for the second messenger system. So look at this phospholipase C react on phosphoinositol, and they are broken down this phosphoinositol into two parts, the IP3 and diacylglycerol. So this two part actually very important. These two things are very important for the second messenger system, give the signal to the cell. So look at this, the whenever the some ligand or some stimulus come to the cell membrane receptors, the phospholipase C activate, and phospholipase C activate the protein kinase, which ultimately activate the phospholipase A2. So this is the second messenger system, diacylglycerol and IP3, which is mainly activated after the activation of phospholipase C. So this activate the diacylglycerol and IP3. So phospholipase A2 activate, and its phospholipase A2 break down this arachidonic acid, and this inflammation process will started. So this is how this the importance of phospholipase A2 and phospholipase C. Now, coming to the surfactant, first we have to understand how do lipids behave in aqueous environment actually. So this is you can see this phospholipid are amphipathic molecules. They aggregate to hide their hydrophobic tails. They are hiding their hydrophobic tails from the water inside and expose the hydrophilic head outside. So the phosphate part they are showing on in front of the water, whereas the tail is hiding there. Major functions of phospholipids are enter in the structure of cell membrane, the component of lipoproteins. Blood clotting is very important function. Phospholipid as a component of bile. In bile makes some cholesterol as well. And the major function which we are now going to discuss about the dipalmitoyl lecithin, which is act as a lung surfactant, which prevent the alveolar collapse and allow the air passages. So this is all about the phospholipid. Before coming to surfactant, we have to first discuss about the sphingolipid as well, which is also one of the phospholipid. Very small amount the sphingolipids are present in surfactant, but we have to discuss this. Sphingolipids contain the sphingosine as an alcohol. Fatty acid is attached to amino group of sphingosine, and phosphatidylcholine is attached. And the major source is brain and nerve tissue. As I told you, very small amount is also present in the surfactant as well. So this is sphingosine, not the glycerol. The sphingoalcohol, sphingosine is present here with one fatty acid and phosphate group, and one choline is attached. This is the structure of sphingomyelin. So the synthesis is mainly have four major steps of the formation of sphingomyelin or sphingophospholipid. Number one is the fatty acid, which is called as palmitic acid, is activated and to form the palmityl CoA, and this palmityl CoA react with the serine to form sphingosine derivative, and after that is formed the sphingosine base. And because you know this base is very important to formation of a phospholipid, and this sphingosine base and react with another fatty acyl group and to form the ceramide, and then this ceramide is react with the choline part, the nitrogenous base, and form the sphingomyelin or sphingophospholipid. And this is what the palmitoic acid, the fatty acid, combined with serine to form the one compound, intermediate compound, the ketosphingosine, and the further reduction and oxidation process occur and form the sphingosine. This is one types of alcohol like a glycerol, and this glycerol combined with one of the fatty acid, fatty acyl group, and this fatty acyl group with sphingosine form the ceramide, and finally the nitrogenous base, the choline will attach as there and form the sphingolipid or sphingomyelin.